Hello, I am Ivan Carrera, and this is my work representing cellular lines with support vector machines and text processing at the International Conference on Bioinformatics, Computational Biology, and Health Informatics. I'm a professor at the Department of Informatics and Computer Science at Escuela Politécnica Nacional del Ecuador. I'm also a PhD student at University of Porto. Inés Dutra and Eduardo Tejera, my co-authors in this work, are my advisors. My research in my PhD is using machine learning and artificial intelligence for discovery of interactions between biological entities, in this case, cellular lines. The contribution of this work is a method for representing cellular lines. The main problem for predicting cell line interactions with chemical compounds is the lack of a computational representation for cell lines. Current approaches compare gene expression profiles, which are frequently unknown. We present a method for characterizing cell lines from scientific literature, so it's no longer necessary to know the gene expression profiles. We retrieve and process cell line-related scientific papers, perform a document classification algorithm, and then obtain a description of the information space of each cell line. We have successfully characterized a set of 300 plus cell lines. The pipeline for obtaining the cellular line representation is this. First, we identify cellular lines. We work with Cellosaurus database. We then retrieve the abstracts from PubMed. Then we tag the data from the corpus. Here is an example of the abstracts for a particular cellular line. We have all the documents, and all the documents have a certain metadata, and they are all tagged with the cell ID. This is going to be the class for our classification algorithm. Then we transform the data from this text to TF-IDF representation. Once we have these vectors, we perform feature selection in order to lessen the complexity of our algorithm. Then we perform the document classification algorithm, and after that we have the cell line description, which shows like this. In this example, each row, each line represents a particular cellular line, and each column represents one of the dimensions of the support vector. The coefficients of the support vector represent the terms of the TF-IDF. The results of our work show that this description allows to classify correctly cells. We divide our data into train and test datasets. And we select line with documents above a certain minimum and see that with more information, we can achieve better performance. Support vector data description is a method for data characterization based on support vector classifiers, obtaining a spherical boundary around a dataset in order to describe it. In document classification, each document is represented as a vector of TF-IDF term frequencies. Each term corresponds to a vector dimension. We found that for this problem, contrary to what was expressed by this work, reducing the number of these dimensions does not affect the performance of document classification, as shown is in figure before. We presume that the corpus is diverse enough not to be affected by removing dimensions. In this work, we have been able to characterize cellular lines using their scientific literature. In performance, we achieve more or less 90% for trained dataset and 70% for test dataset for micro average precision for classes of 60 plus documents. The limitation is that for high accuracy, we need cell lines with large corpus, making our method less general. Future work should test the value of the representation of cellular lines in the drug discovery process. These are my references. Thank you.